leaves the nucleus of the cell. It is up to making messenger RNA to begin making the protein. This is where my messenger RNA pairs to the bases. This is where I would get A to U, T to A, G to C, C to G, A to U, T to A. Now, every 3 bases on the DNA is a code. Every 3 bases on my messenger RNA is the codon. That strand moves out, ribosomes attach, tRNA will carry 3, so let's see, U, G, whoops, y'all should have corrected me on that. My U, what did I do wrong, y'all? I did something wrong. No, I didn't. ATGC, ATGC, so A goes to U, T still goes to A, G goes to C. So now U goes to A goes to U, C goes to G. So now this would have a particular amino acid attached. And it would come in and match up to these 3 bases. And as it does, it brings in its amino acid. This one, the tRNA, would be CAU, and this one would have an amino acid. And it would come in. And as it does this, that ribosome is moving all the way down, and it brings in its amino acid and forms the bond. So as we continue this for anywhere from hundreds to thousands of base pairs long. So for every 3, an amino acid gets to come in. So the next one could be like this. The next one could be like this. Do you see where I'm going? So we simply amino acid, amino acid, amino acid, amino acid, and when it finally gets to a stop codon, it stops. Ribosome breaks away from it. I now have a single strand of amino acids. And that single strand of amino acids is the beginning of my protein.
That would be a primary structure.
That would be my primary structure. And from that point, that primary structure can twist or bend, which is my secondary, which can fold in on itself, which is, well, that's secondary, and then it can fold in, and I get my tertiary, 2 or more together, quaternary.
And those little letters, the bases, they are the nucleotides.
They are the nucleotides. Yep.
The 3 little bases.
Yep.
That's
The A's, T's, G's, C's, and U's. Those are the bases, those nucleotide bases, which is also on the test. So pretty much, this is what we're going to see in the presentation, just in graphics. This is showing where my messenger RNA strand, and remember I said the ribosome gets to simply move down. So 3 bases, and then we'll see the next 3 bases. We'll see the next 3, and each time for every 3, an amino acid comes in, an amino acid comes in, and it just continues. We form the bond, moves the next 3, amino acid, woo, until we get a whole string of them. Does that make sense? And once we have the string of amino acids, that's our primary structure. I don't know if this is going to play. Let's see if it'll play. Just may not have any audio. Because no speaker system.
Translation is initiated by formation of an initiation complex consisting of a fuller ribosomal subunit. The first amino acid, tRNA.
I apologize for the audio, but you can at least see the pictures.
Subunit that joins the complex. Proteins called initiation factors are also involved, but are not shown. The 7S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon, and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain.
Do you see how they're trying to show us that that messenger RNA can be hundreds of thousands of bases long? And so for every 3, a transfer RNA drops in, bringing an amino acid until it's told to stop.
Is the peptide bond formed in the ribosomal unit or once
Yes.
it exits?
It helps. The ribosomal unit will help form that
So
peptide
when it
bond.
goes from like the
From
A
the,
site to the P site,
Yes.
and then that's when it's made. It's not when it as it leaves.
no.
All right.
In other words, the ribosome will help to form that peptide bond. Does that kind of help explain what I was trying to draw? Okay, every 3 bases, 3, 3, 3, and you got to remember, every protein that is made by the body has a gene with its number of bases. That's why it is so important for this process to occur correctly. Because if there is anything in this process, like a base being incorrect, where there's a U and there's a C, that protein will now be incorrect. And it will not do the job it's intended for. So at this level, it's very important for this process to occur correctly. Because what do we say about the structure of a protein? It determines what? Function. If so much as 1 base is wrong, the protein is wrong. If an entire codon is left out, it's wrong. If an exon and intron doesn't work correctly, it's wrong. So it's very important for this process to occur correctly. So it is a process that's occurring in nanoseconds. We're talking in just seconds of time that this actually occurs. And look at how nicely it does it. It's almost 100% correct every time.
Okay, so when you say one thing's wrong, there is the mutation, right?
Yes.
So do these mutations, like, do they keep happening in that one cell, or like it can just be that one?
Now, in some cases, the cell will immediately correct it, and therefore whatever was wrong, the cell itself, for example, if it's something that's going to affect the cell overall and the cell catches it, so to speak, the cell will kill itself. It'll carry out apoptosis. And because the other cells around it are carrying the job correctly. In some cases where if the mutation occurs and the cell doesn't correct it, in the grand scheme of things, that means the next time it's made, it's going to do it again, and it's going to do it again, and it's going to do it again, and eventually something goes wrong with that tissue, which of course could be something going wrong with the individual. In some cases, when these don't correct, the whole especially like during times of development and the sperm beating and that sort of thing, the offspring will die. So it just depends on if it's a mutation that the individual can still survive. Like for example, people with trisomy 21, Down syndrome. This is where things didn't go right in the cell, but the individual is still alive. Yeah. But in some cases it does. It can actually kill the cell itself and be done, and other times the individual can live with it, other times the individual dies. So the process needs to be very precise, and our bodies do that on a regular basis, and it does it very precise. So when we have this process, okay, in making proteins, we are simply working, this is basically the time frame the cell is doing its job, okay? So, we do regulate the expression of these genes, meaning, okay, we are going to make sure that not every, and this is true, in your entire genetic makeup, do you happen to know about how many genes the entire human population has as a whole? And of the genes, okay, in the entire human population as a whole, there are about 50,000 genes. You don't have all 50,000, okay? You may have 30,000, but not all 30,000 genes that you have are expressed. Some genes that you have can stay turned off your entire lifespan. Some, if you get exposed to a particular chemical, because that's what makes the genes act, okay, is chemical information. Sometimes you can have genes that if you're exposed to that chemical during your lifetime, that gene gets activated. And it now begins to do stuff it shouldn't. For example, cancer. Okay? We have a lot of cancers now that we didn't have back in the 1800s, early 1900s, but you look at all the chemicals we're exposed to now. And they're turning on these genes.
Is that kind of like the
relate Allen piece to?